All right. Today is day 2, and our topic today is biochemistry. And what we're going to focus on are the 4 macromolecules. So now that you know what biology is, what the characteristics of life are, and how to conduct an experiment, collect data, analyze data, and come up with a conclusion, we're going to go ahead and launch into microbiology, which is what this class is about, just microbiology. And so before we get to our topic of cells, we need to really dive into the molecules that make up cells and that make things living. And so those are called macromolecules. And so the Earth was formed about 4.6 billion years ago. So we're going to talk about the origin of cellular life. Where did it all come from? And for about 500 million years, the Earth was continually bombarded with chunks of rock and ice in the solar system. Following this heavy bombardment, the early Earth atmosphere contained. And so this is a theory. We obviously can't, we didn't have a camera set up or anything like that. We didn't have anyone like taking notes. But through a series of experiments, it has been determined that early Earth atmosphere contained water vapor, also known as H2O, nitrogen, N2. We know nitrogen is one of the diatomics. It can't be alone. It has to be with another nitrogen. Carbon dioxide, or CO2, methane, CH4, and ammonia, NH3. What I want you to notice about this list is that oxygen, O2, is not there. And so it is believed that the early Earth atmosphere and the very first life forms did not use oxygen. And so another word for that would be anaerobic. It is believed that early Earth forms were anaerobic. So I want you to remember that for our cellular respiration chapter or unit that we're going to go over in the coming days. So these were what was believed to existed in early Earth atmosphere. So how did life, and those are some harsh chemicals. I mean, ammonia and methane, oh my gosh, those are pretty harsh. So how did life arise from such a harsh environment? So you have these two scientists that designed a model of what the conditions of Earth were like at this time, and their names were Miller and Urey. So those were their last names. And so the thing that they set up, this apparatus that they set up, was called the Miller-Urey apparatus, also known as the Miller-Urey experiment. So the Miller-Urey apparatus or the Miller-Urey experiment. And there was two scientists. So hopefully you got that down. And this is what it looked like. Looks pretty cool. It almost looks like it reminds me of like an organic chemistry setup where you're like distilling something, because distillation involves evaporation and condensation down into collecting the condensate. But they weren't distilling. What they were doing was they were simulating the 3 important conditions on early Earth. And so it was believed that there was a lot of lightning. And so they had electrodes up here. So if you can look at my cursor, a lot of electrodes that are simulating lightning. Heat and gases that were released by volcanic activity. And so you have all of your gases right here, your ammonia, hydrogen, methane, and then water vapor. So these 3 things, these 3 important conditions, lightning, heat and gases from volcanic activity, and water vapor. And so what they found was if they boiled water, water vapor rises. It is going to mix with lightning and volcanic gases. So water vapor mixes with volcanic gases in the presence of lightning and then condenses down, gets cooled off, and collects in this flask right here. Okay, so that was the experiment. We're basically mixing water vapor with volcanic gases in the presence of lightning. All right, so what happened? After 2 weeks, 10 to 15% of the carbon had been used to form organic compounds. So what was formed in this flask right here, they found to be organic compounds. In particular, they found amino acids that had collected in that flask, which was so awesome and just so revolutionary that they found that they could make amino acids out of those simple compounds and just water vapor and lightning and stuff like that. And so it was assumed that this is what early Earth looked like and that the conditions were there in order to make organic compounds. That could happen. And so what purpose did the lightning serve? And I know I keep going back, but what purpose did the lightning serve? Well, the lightning was there to break bonds and then bonds reformed. And so you have your carbon source. You remember carbon dioxide was also there. You have your oxygen source, which came from water, H2O, and then you have your hydrogen sources, and then your nitrogens and all of that stuff. So you'll see where all of those come into play when we talk about the different macromolecules. All right, so hopefully that kind of made sense that these simple organic compounds could have been produced. That the ones that could have been produced were proteins, lipids, carbohydrates, and nucleic acids. And so those are our 4 macromolecules. Those are the 4 molecules that must be present in order for something to be living. All right. And so what we're studying today is biochemistry, and in particular, organic chemistry. Organic chemistry is, and if some of you have, whether research or scientists or physician parents, I want you to ask them what their hardest class was in undergrad, and I bet you anything they will say organic chemistry. It is so very difficult. I made it through though, so it's manageable and it's doable, but it is one of the hardest classes. But for this class, it's really not. It's actually pretty interesting because we talk about the organic compounds that are found in food. That's always something that's relatable because we all eat. So organic chemistry is going to be the study of carbon compounds that make up living organisms. So the 4 macromolecules: carbohydrates, proteins, lipids, and nucleic acids. Why is carbon so special? Is because it is considered the backbone of all organic compounds because it has this unusual, it's small. And so if you can kind of picture where it is on the periodic table, it doesn't have, I think it's in the second row. Yeah, it's in the second row, so it only has 2 rings, right? Which makes it small. And then it has the ability to form 4 covalent bonds. So it's very versatile. It can take on, carbon compounds can take on a lot of different shapes. And so here's just a lot of different compounds that can be formed with carbon. So it's very versatile. So the most common organic molecules that are found in living things are really, really big. They're like huge, and they're made up of mostly carbon. There's as many as 1,000 atoms of carbons and other atoms bonded together to make up these macromolecules. And due to their size, they are referred to as macromolecules, so that's the word macro. So what you're seeing right here is a macromolecule. It's a carbohydrate, and it's called cellulose, and I'm going to talk about it in just a minute. I don't want to spoil it and give it away, but just in case you're wondering, it is huge. Cellulose, which is the carbohydrate found in plants, it's what you can't digest. It's what cows can digest because they have a special bacteria in their gut, but it's what you can't digest. So it's also known as fiber. Whenever you're looking at nutrition label and you see fiber, that's actually cellulose that they're talking about. All right, so we are going to start with carbohydrates. Then we will discuss nucleic acids, then lipids, then my favorite and what I consider the most important one is proteins. All right, so starting with carbohydrates. The way that you are going to recognize if something is a carbohydrate is if it is made with carbon, hydrogen, and oxygen, and it's going to be in a 1:2:1 ratio. And our classic carbohydrate that we will mention over and over and over again is glucose, C6H12O6. And I wish I knew how to do the little pen marker thing, otherwise I would write that for you, but I don't know how, and I haven't really learned how. I probably will, but I'm not at this moment. So a 1:2:1 ratio of carbon, hydrogen, oxygen. Glucose is our classic example, C6H12O6. Glucose is also known as a monosaccharide. So anytime you put a carbohydrate into water, into an aqueous environment, it's going to form what we call a ring. And I'm going to show you a picture of that in just a minute. If one ring is formed, it's called a monosaccharide. Mono meaning one, saccharide meaning sugar. And here are our examples. Glucose, So this is one ring. And if you counted up all the atoms, it would still be C6H12O6, galactose, fructose, and then we have two others, deoxyribose, which is the sugar that's actually found in DNA, and then ribose, which is the sugar that's found in RNA. So as you can see, the carbon, hydrogen, oxygen are in a 1 to 2 to 1 ratio, and they form rings, either hexagons or pentagons. And then these are some of our monosaccharide examples, meaning that they're just one ring. Okay? And so glucose is what's going to be found in your blood sugar. Galactose is found, galactose helps make up lactose, which is found in milk. Fructose is found in fruit. And they're all for structural and energy storage. Right? And then ribose and deoxyribose are found. So ribose is in RNA, deoxyribose is in DNA. And that's easy to remember because ribose starts with R, deoxyribose starts with D. All right. So if a monosaccharide is one ring, then a disaccharide is going to have two rings. So it's like a double sugar. So it's two monosaccharides bonded together. Sucrose, which is also known as table sugar, which is what you used yesterday to do your yeast experiment. It is made up of a glucose and a fructose molecule bonded together. Maltose, which is malt sugar, is a glucose with a glucose, and lactose is a milk sugar, like I said a minute ago, is a glucose with a galactose. I don't really need you to remember any of those except for sucrose. I think that's an important one to remember. Sucrose is a disaccharide. It is made up of glucose bonded to fructose. That way you can demonstrate that you know what a disaccharide is. Two rings. All right. So if mono means one, di means two, then poly is going to mean more than two. And it actually is going to mean a lot. Like not 3, 4, like hundreds. All right. So a polysaccharide is an example of a polymer. Has a large number of repeating similar units bonded together. Right? Polysaccharides in living organisms are going to fill one of two roles, providing structure or energy storage. And we have a few examples of these from animals and plants, and there is one for fungus, but I don't have it on here, but in AP Bio you'll learn that one. I'll just go over 3 examples. All right, the first one is starch. Starch is the energy storage polysaccharide in plants. It is a long branching, so what I'd like for you to write down on this picture is starch, energy storage in plants, and it's long branching. See how they're branching? They're not in rows. They're branching off of each other, and this shows you an up close picture of the rings, and then this shows you what the whole thing would look like. It almost looks like a tree, right? So long branching. Students will get confused when I ask them what is the energy storage in animals, and I give them a multiple choice. So I'm probably going to give you this on your quiz tomorrow. I give them a multiple choice and I list all the polysaccharides, and they want to tell me that starch is energy storage in animals, and I say no. We eat starch, but we don't store it as starch. We store it as glycogen. And you're like, but that looks exactly the same. But it's not exactly the same. There is some different branching to it. So if starch is the energy storage in plants, and it's stored in roots or structures like potatoes, glycogen is the storage polysaccharide in animals, and it is located in your liver. So it's nearly identical to starch, except there's more branching. All right. And so humans and other vertebrates are going to store glycogen in their liver and in your muscle cells. So it's like your savings account, basically. So if you were to go on a low carb diet, or if you were to run out of food, and I don't know if it was a nuclear winter and we ran out of food, after a while, your body would then tap into the energy storage in your liver, giving you the kind of that last reserve. It's like your savings account. And then it's stored in your muscle cells for really, really quick bursts of energy if you need to run from a lion or something like that. Right, so this is the energy reserve utilized for quick bursts of activity. All right, so that's starch and glycogen. Okay, cellulose. Cellulose is a structural polysaccharide that's part of the cell wall of plants. And it is not branching. It is in like rows or sheets. I actually would write down the word sheets. So this is what allows the plant to be sturdy and rigid. And this is what makes up the cell wall, and it keeps the cell wall from bursting when it fills up with water. It's similar to starch in that it is also made out of glucose molecules. So all three of these, y'all, starch, glycogen, and cellulose, the one thing they all have in common is that they're polysaccharides that are made up of glucose molecules. They are all made up of glucose molecules. So what's the difference is that how those glucose molecules are arranged. So in starch, the glucose molecules are branching. In glycogen, the glucose molecules are bonded together and there's more branching. Cellulose, the glucose molecules are all bonded together, but they're arranged in sheets. And so that's what makes the difference. So we are unable to digest cellulose, like I said before. That is your fiber, also known as roughage. So they say iceberg lettuce has no nutritional value, and I argue that. Iceberg lettuce has a lot of fiber, and fiber is really good for you in keeping you regular, but it's also really good for you in preventing cancer. And when we get to our cancer chapter, I think that's next Friday. I will, or it's day 10, if you're counting days, it's day 10. I will talk to you about why that's so important in preventing cancer. So we don't digest it, but cows do. They have a bacteria that lives in their gut that helps them digest cellulose. And what's the reason why we can't digest cellulose? It has to do with the way that they're bonded together. And I really don't get into that detail in cell bio, but in AP bio, I do get into these alpha linkages and beta linkages, and we are unable to break those beta linkages. And so that's why we can't, or the enzymes in our stomach cannot break those beta linkages, and that's why. All right. And that is carbohydrates. So carbohydrates are also known as sugar, but they don't always taste sweet. I could give you a cracker, and that wouldn't taste sweet. It'd be real like starchy. But sometimes if you put a saltine cracker in your mouth and you let the enzymes, you have amylase in your mouth, you let the enzymes digest it, a saltine cracker can actually take on a kind of a sweet taste. All right, our second macromolecule is nucleic acids. We have two nucleic acids, and that's DNA and RNA. And we do have lots of different days where we're going to talk about DNA, RNA, and what those are used for in particular. Today, our only purpose is to just see what atoms are they made up of and what structure do they take on. So carbohydrates have CHO in a 1 to 2 to 1 ratio. How you're going to recognize nucleic acids, and I would write this down, is you have C, H, O, N, and P. CHONP. All right, this is the only macromolecule. Well, I take that back. This is the only macromolecule that is going to have a phosphorus every single time. All right, and so nucleic acids are made up of repeated units called nucleotides. So this is the monomer of a nucleic acid. This is the unit of a nucleic acid. And so in order to have DNA or RNA, you're going to have a bunch of these things bonded together. And they have 3 parts: a phosphate group, a nitrogen-containing base, and just to give you a little preview, this nitrogen-containing base is where you get that ATCG that you probably learned about in middle school. And then you have a sugar. And so remember, for DNA, it's deoxyribose, and for RNA, it's ribose is what the sugar is going to be called. All right. DNA is made up of 2 strands of nucleotides. And when we talk about the structure of DNA, it'll make a little bit more sense. They run anti-parallel to each other, and so it causes them to twist around. So DNA is made of 2 strands of repeated nucleotides. They twist around, forming a double helix. The nitrogen bases here are going to form hydrogen bonds. So anytime you see a dotted line, that's a hydrogen bond, and I'm going to get into why it's a hydrogen bond a little later. I'm just giving you some structural information now, and when we talk about DNA replication, that will make more sense. Really, in case you just already want to know, it's hydrogen bonds because DNA has to be pulled apart so that it can be copied. All right, and then RNA is going to be a single-stranded molecule, and I have a little picture for you on your notes. A single-stranded molecule. There's no hydrogen bonding going on or anything like that. And then the other difference is that DNA is going to have the nitrogenous bases ATCG, and RNA is going to have AUCG. But I definitely wouldn't get too caught up in those details. What I really want you to know is, how do you recognize nucleic acid, CHONP, and what are the 3 parts of a nucleotide? And then DNA is double-stranded, RNA is single-stranded. All right. Third macromolecule, lipids. Lipids, like carbohydrates, are made up of carbon, hydrogen, and oxygen. So you have CHO again. Except this time, it's not in a 1 to 2 to 1 ratio. This time, you have way more carbons and hydrogens than you do oxygens. So many carbons and hydrogens and a couple of oxygens. So why is that significant? It is significant because if something is made up of mostly carbons and hydrogens, it is considered a hydrocarbon. I know I just switched the words. It's considered a hydrocarbon. And since carbon and hydrogen do not have enough of a difference in electronegativity to pull electrons towards one atom or the other, it is considered non-polar. So there's no partial charges. So lipids are going to be waxy, fatty, and most important, what I want you to know about lipids is that they're hydrophobic. Hydrophobic. All right, so they are the only class of molecules that don't form polymers. They're hydrophobic because they cannot dissolve in water. So if you think of if you poured vegetable oil in a glass of water, what would it do? It would float to the top. And then, like I said just a minute ago, the reason why they're hydrophobic is because they are made up of mostly carbon and hydrogen. So they're hydrocarbons. There is not enough of a difference in electronegativity between carbon and hydrogen to pull electrons towards one or the other. So they are considered non-polar. There's no partial charges, so they don't like water. A little chemistry in there for you. So if you haven't taken chemistry, I think you do that in 8th grade chemistry. Actually, I think you talk about hydrophobic and hydrophilic. I hope so. If you don't, then let me know. I can explain it again. All right, we have different types of lipids. So we don't have any monomers or repeated monomers or polymers or anything like that. We just have 3 distinct types of lipids that we talk about. The first one are called triglycerides. Triglycerides, what I want you to think of is just fat. Triglycerides are fat. And that's easy to remember because if you look at this picture right here, that's super fat. I mean, that is so many carbons and hydrogens, it's ridiculous. All right? So that's how you recognize a triglyceride. And so you have a glycerol right here. And then these chains are called fatty acids. They're fatty acids because it's just carbons and hydrogens. Long chains of hydrocarbons. Fatty acids are going to vary in 2 ways, the length and the presence of double bonds. All right, and when we talk about double bonds, we're talking about the double bonds between carbons. A saturated fatty acid is going to have the maximum number of hydrogens possible, no double bonds. So this picture right here would be a saturated fatty acid because you don't see any double bonds. As many hydrogens as could bond to the carbons as possible are there. That is a saturated fatty acid. A saturated fat, is going to be solid at room temperature. An example would be lard or butter, my favorite. They have a straight shape, and so they pack together more tightly, and that's why they're solid at room temperature. Unsaturated fatty acids have 1 or more double bond between 2 carbons. And so when they have 1 or more double bond between 2 carbons, it causes the tail to bend or kink. We call it kinking, K-I-N-K. So the tail bends or kinks because there's a double bond between carbons. And then there's a curve shape. And so the curved shape of this tail right here keeps them from packing together so it stays liquid at room temperature. And our example for this would be olive oil. All right, our second type of lipid are phospholipids, and phospholipids are really important because they're going to make up the cell membrane. So this one was going to carry over in future discussions about the cell membrane. It has a phosphate head and two fatty acid tails. So that's how you can recognize it, is that it has carbon, hydrogens, oxygen. So it has those hydrocarbon tails right here. And then it has a phosphate head. So what makes phospholipids so unique is that they are considered amphipathic, meaning they have both hydrophilic and hydrophobic properties. So we already talked about why the fatty acid tails are hydrophobic, and that's because they're made up of mostly carbons and hydrogens. They're nonpolar, there's no partial charges, therefore they don't like water. But this phosphate head, we know phosphates have charges on them. Anytime you have something that has a full or a partial charge, it's going to get along with water really, really well because water has partial charges, right? And so we consider this phosphate head to be hydrophilic and these two fatty acid tails to be hydrophobic. So it has both properties. And so that's going to be really important when we look at the cell membrane. So I would just hang on to that thought. So when they're added to water, the hydrophilic part faces the outside. So pretend that this is surrounded by water. If it's hydrophilic and it loves water, it's going to be facing water. If it's hydrophobic and it has no partial charges, is nonpolar, it doesn't want to be around water, it's going to face the interior. And so this is how the lipid bilayer of the cell membrane is actually set up, is this right here, right? And we will go over that in detail in the upcoming days. But I just thought it was important to mention that the cell membrane is made up of two layers of phospholipids that turn in on each other with the hydrophobic tails turned towards each other, which is pretty cool. And that contributes to the entire function of the cell membrane, which is to let some things in and not other things. Pretty cool. All right, the third type of lipid, and I think this is the final type, yes, that we are going to talk about, are called steroids. So as you can see here, this molecule looks very different than the other ones that we've talked about. We don't see any fatty acid chains. We don't really see a whole lot of hydrocarbon going on here. And students tend to get a little bit confused, especially those that don't have quite as much chemistry experience. What I want you to know is when you see a ring with no letters, it's a hydrocarbon. What chemists have done is they've just eliminated writing all the carbons and hydrogens because they don't see any point. If we can just draw a hexagon and you just assume those are carbons single bonded to each other and then there's hydrogens hanging off of there, then you can eliminate a bunch of mess in the picture. So we see 4 rings fused together, and that is the hallmark of a steroid, is 4 hydrocarbon rings fused together. Right, so they have a different structure, like I said. 4 interlocking, I like to call them fused rings, not so much interlocking, fused, because fused means that 2 rings are sharing one side, like this right here. Okay, and so your steroids are going to include cholesterol, cortisol, testosterone, and estrogen, and progesterone. So when you think of cholesterol, you're like, oh, that's what's found in eggs. That's what my grandparents has to not eat so much of because they have high cholesterol. So we tend to think cholesterol is a bad thing, but it's actually not. Cholesterol plays a really important part in the integrity of our cell membrane. And cholesterol is the precursor to testosterone and estrogen, meaning cholesterol is what let's say you eat an egg and the egg has a bunch of cholesterol. That cholesterol then gets transformed into the sex hormones. So cholesterol is needed. Now, when cholesterol is too abundant and it starts clogging blood vessels, yes, it's bad, but a diet with 0 cholesterol isn't necessarily considered that great either. So overall, this is the third type of lipid. It's called a steroid. The way you're going to recognize it is that it has 4 fused rings, and our example of steroids are going to be cholesterol, cortisol, which is the stress hormone, and then sex hormones. And so you would automatically assume all steroids are going to be hydrophobic as well because they're made up of hydrocarbon rings. Okay, and that's all for lipids. Let's move on to the fourth and final macromolecule, which is proteins. The one that I consider the most important, because it's not that they're the most important because they're the only one that we need. We need all of them. It's the one I consider most important because it carries over to all of our other topics up until the last day. We're going to talk about genetic diseases. Genetic diseases is an error in DNA that ends up messing up a protein, and that's why the person has the disease. So proteins carry over into so many different topics. So that's why I consider it to be the most important. All right, proteins, and I'm going to try to move this down so you can see the thing about hemoglobin down there. Proteins are made up of chains of amino acids. So the monomer of a protein is called an amino acid. The way you are going to recognize a protein, so write down these letters because I don't see them on this slide. C, H, O, N, S. Chons. CHONS. Proteins are the only macromolecule that have sulfur. All right. And so they're made up of chains of amino acids, and you have 20 different types of amino acids, or 20 different amino acids, and they can be arranged in an infinite number of ways. A typical protein is going to be about 400 amino acids long, and that can vary, but I would say just your average typical protein is 400 amino acids long, and you have 20 different amino acids to choose from. The sequence of the amino acids is going to determine the shape of a protein, and the shape of the protein is what's going to determine its function. And so down here are just two examples of a couple of proteins that are really important. Keratin. Keratin is really linear. Keratin is a structural protein that's found in your skin, hair, and nails. Keratin is usually considered, when you have a keratinized cell, it's usually a cell filled with keratin, and it's actually a dead cell. But it's very important for structure. All right, hemoglobin. Hemoglobin is what's considered a globular protein, and it just looks like a big old glob it's like they took some strips of paper and clumped them up together to make this protein. But I can tell you, even if it looks like a glob mess, the folding is actually very, very specific in how this protein is going to function and where it's going to fit. So let's talk about that for a second. I think I have the amino acid chart. I don't have the amino acid chart here, but 20 different amino acids. Oh, you have the amino acid chart on your paper, sorry. So on your paper, you see the 20 different amino acids. You don't need to memorize any of those. I had to memorize them in school. I remember studying the flashcards for them, and I never used that knowledge because I never had to memorize it. Why memorize amino acids when you can just look them up? So you have 20 different amino acids. Remember, each protein is about 400 amino acids long. So when you look at the chain of amino acids that are bonded together, that's called the primary protein structures, the chain of amino acids. What happens then is that the amino acids then start interacting with each other. And when they start interacting with each other, they start forming hydrogen bonds between each other, and they will take on shapes called an alpha helix and a beta pleat. That is called secondary structure. So these are like the steps of protein folding. First, you bond your amino acids together. Then the amino acids start interacting to form alpha helix and beta pleats. Then the amino acids start folding in on each other even more to form this glob. That's called a tertiary structure. Then when multiple tertiary structures come together, that's called a quaternary structure. Some proteins are functional at the tertiary structure. Some proteins are not functional until they reach a quaternary structure. So hemoglobin is going to require 4 different polypeptides that have been folded up in order to be functional. Keratin and collagen are going to require 3. But there are some channel proteins that are going to only need 1 little glob. All right, so hopefully that makes sense. I'm probably going to go over protein folding in Zoom tomorrow. Okay, and so just to give you an example of protein folding and how important it is, morphine is a drug, And so this is how some drugs are actually made. So endorphins are proteins that are naturally produced by your brain to reduce pain and to stimulate feelings of euphoria. So when a person is a runner and they experience a runner's high, that's actually their body dealing with the pain of running, and it actually gives them an endorphin high, kind of similar, a little bit similar to morphine. So that's why runners get, and I run, and you like crave running after a while because that feeling is so amazing. So an endorphin is a natural painkiller that's produced in the body that fits into a receptor. And a receptor is a protein that's in the cell membrane. So it fits into the receptor and it sends a signal and it gives you that feeling. So morphine is actually a drug. So it's exogenous. It's not made within the body, but it is put into the body to mimic an endorphin. And so when a person is in pain from surgery, what they can do is give them a hefty dose of morphine, and it's almost like endorphins, but like maxed out like times 1,000. And that's why a person won't feel pain, is because they're shaped similarly in a certain spot. And this is the spot here, this is the spot here. And so the protein is folded similarly in these two spots, can fit into that pain receptor, causing the person to not have pain. Great. Proteins are going to account for 20% of total body weight, and they're going to perform a variety of really important functions. And so this is a reason why it is going to carry over into a lot of different topics. So protein functions are going to include structural support, movement, transport, you can just write down transport, buffering against changes in pH, so like in the blood, so it can work as a buffer, regulation of chemical reactions, and then control and defense, like antibodies. So you might want to pause here so you can get those written down. Enzymes are one thing. So if you look back here, regulation of chemical reactions, I'm going to talk about that for a second. Enzymes are special proteins that catalyze or speed up the rate of a reaction in the body. And so the reactants are called the substrates, and the products are what are produced following a reaction. And so we are going to talk about enzymes in greater detail in a few days, but I just wanted to mention that the folding of an enzyme, because an enzyme is a catalyst for a chemical reaction. So let me give you an example. For digestion, you have an enzyme in your mouth called amylase. And so when you eat a cracker that has a bunch of carbohydrates like amylose and other carbohydrates, amylase is going to break those carbohydrates apart. Same thing with lactose. Lactose is in your digestive system and is going to break, lactase is in your digestive system and it's going to break lactose apart. That's why people who are lactose intolerant are lactose intolerant, because they lack the enzyme lactase that helps digest it, right? So enzymes are important. They're important for chemical reactions. The folding of an enzyme is really, really important, because if it becomes unfolded, then it loses its function. And so you can have an active protein, and then if it unfolds, it is inactive. And then if it becomes permanently denatured, it cannot reconfigure itself. So an example would be when you fry an egg. So when you think of when you crack an egg, it's like clear, the protein, the clear part, egg white is what you call it. It's clear and it's actual protein, but then when you cook it, you're denaturing or unfolding the proteins permanently, and that's why it looks the way it does when you denature it. So the folding, what I want you to get from this is that proteins are important. They serve so many functions. I will talk about proteins like over and over and over again because they're that important. And if they become unfolded for any reason, they will become non-functional. And we will continue enzymes on Friday. All right, so that is all I have for you for macromolecules. And if you can go ahead and answer the summary question so I know that you understood what we talked about, and then I will see you guys later.